Molecular basis of inheritance. So the context is the DNA, the search for genetic material, RNA world. So the DNA, the search for genetic material, RNA world, replication, transcription, genetic code translation, regulation of gene expression, human genome project, and DNA fingerprinting. So these are the contexts we are going to study. So in this chapter, what is okay? So the Mendel gives the factors. Okay, Mendel gives the name, that is factors, term factor. Now DNA, deoxyribonucleic acid, is the genetic material, at least for the majority of the organisms it is DNA, right? Now the nucleic acids are polymers of nucleotides. Now deoxyribonucleic acid, DNA, and the ribonucleic acid, RNA, are two types of nucleic acids found in living systems. Now DNA acts as the genetic material in most of the organisms. RNA, though it also acts as genetic material in some viruses, mostly functions as a messenger. Okay, so in some viruses, but they mostly functions as a messenger. So RNA has additional roles as well, like adapter and all that, mRNA, okay, and then tRNA, then rRNA, that is okay. So it functions as adapter, that is structural, and in some cases as a catalytic molecule as well. So it is genetic material in some viruses, but although it is majorly the messenger. Okay, now the structures of nucleotides and the way these monomer units are linked to form the nucleic acid polymers. Okay, we are going to study this. We are going to discuss the structure of DNA, its replication, the process of making RNA from DNA, the process of making RNA from DNA, that is transcription, the genetic code that determines the sequences of amino acids in proteins, the genetic code that determines the sequences of amino acids in proteins, the process of protein synthesis, that is translation, and elementary basis for their regulation. Okay. Okay, the structure of the most interesting molecule in the living system, that is the DNA. Okay, and then the most abundant genetic material is the DNA, right? Now the DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as number of nucleotides or a pair of nucleotide referred to as the base pairs. What are the base pairs? These are nucleotides, a pair of nucleotides, right? Now this is the characteristic of an organism. For example, a bacteriophage known as phi into 174 has 5386 nucleotides. Bacteriophage lambda has 48502 base pairs. Phi 174 has 5386 nucleotides. Bacteriophage lambda has 48502 base pairs. Escherichia coli has 4.6 into 10 power 6 base pairs, and haploid content of human DNA, that is 3.3 into 10 power 9 base pairs, right? Now the structure of polynucleotide chain. The chemical structure of a polynucleotide chain, that is DNA or RNA. A nucleotide has three components: a nitrogenous base of a pentose sugar, ribose in case of RNA, and deoxyribose for DNA, and a phosphate group. Now there are two types of nitrogenous bases, that is purines, that is adenine and guanine, pyrimidines, that is cytosine, uracil, and thymine. Now cytosine is common for both DNA and RNA, and thymine is present in DNA, right? Uracil is present in RNA at the place of thymine, right? A nitrogenous base is linked to the OH of one dash carbon pentose sugar through an glycosidic linkage to form a nucleoside, such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, uridine or deoxythymidine, right? Why it is thymidine? Because uracil is not present in DNA. So when a phosphate group is linked to OH of 5 carbon of a nucleoside through phosphoester linkage, right? A corresponding nucleotide or deoxynucleotide, depending upon the type of sugar present. If it is DNA, the sugar present is deoxyribose. If it is RNA, then it is a ribose, okay, is formed. Two nucleotides are linked 3 dash and 5 dash phosphodiester. So two nucleotides are linked through 3 dash to 5 dash phosphodiester linkage to form a dinucleotide. Okay, so more nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has at one end a free phosphate moiety at 5 dash end of the sugar, which is referred to as 5 dash end of polynucleotide chain. Similarly, at the other end of the polymer, the sugar has a free OH of 3 dash carbon group, which is referred to as 3 dash end of the polynucleotide chain. The backbone of a polynucleotide chain is formed due to sugar and phosphate. The backbone of the polynucleotide chain is formed due to sugar and phosphate. Sugar and phosphate, right? Sugar and phosphate. Sugar is ribose or deoxyribose. The nitrogenous bases linked to sugar moiety project from the backbone. So nitrogenous bases linked to sugar moiety project from the backbone. So nitrogenous bases linked to sugar moiety project from the backbone, right? In RNA, every nucleotide residue has an additional OH group present at 2' position in the ribose. Also in RNA, the uracil is found at the place of thymine. 5' methyl uracil, another chemical name for thymine. So thymine is also a 5 methyl uracil. Okay, 5 methyl uracil. Now DNA as an acidic substance present in the nucleus was first identified by Friedrich Mischer in 1869. DNA as an acidic substance present in nucleus was first identified by Friedrich Mischer in 1869. Okay, he called it as a nuclein. Okay, however, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. Then it was only in 1953 that James Watson and Francis Crick, based on the X-ray displacement data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. Now, one of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. However, this proposition was also based on the observation of Irvin Chargaff that for a double-stranded DNA, the ratios between adenine and thymine and guanine and cytosine are constant and equals 1. The base pairing confers a very unique property to the polynucleotide chains. Okay, they are said to be complementary to each other, and therefore if the sequence of bases in one strand is known, then the sequence in other strand can be predicted. Also, if each strand from a DNA acts as a template for synthesis of a new strand, the two double stranded DNA thus produced would be identical to the parental DNA molecule. Because of this, the genetic implication of the structure of DNA become very clear. Okay, the silent feature of double helical structure of DNA. Okay, so it is made up of two polynucleotide chains where the backbone is constituted by sugar and phosphate and the bases project inside. So sugar and phosphate, this is the backbone, and bases are project from inside. Okay, the two chains have anti-parallel polarity. It means if one chain has a polarity that is 5' to 3', the other has 3' to 5', right? The bases in two strands are paired through hydrogen bond, that is H bonds, forming base pairs. Okay, adenine forms two hydrogen bonds with thymine from the opposite strand and vice versa. Okay, similarly guanine is bonded with cytosine with 3 hydrogen bonds. As a result, always purine comes opposite to a pyrimidine. So purine that is adenine, and the purine that is what adenine and guanine, and pyrimidine that is cytosine, uracil and thymine. Okay, so always purine comes opposite to a pyrimidine. So this generates approximately uniform distance between the two strands of the helix. The two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer. Okay, and there are 10 base pair in each turn, right? Consequently, the distance between a base pair in a helix is approximately 0.34 nanometer. 3.4 is the pitch. Okay, the pitch of the helix is 3.4, and 0.34 nanometer is the distance between a base in a helix, right? The plane of one base pair stacks over the other in double helix. This in addition to hydrogen bonds confers stability of the helical structure. Okay, so this is it, right? So this is the thing, right? And this is like the double standard polynucleotide change. You see 5' to 3' and 3' to 5', 5' to 3' and 3' to 5'. Okay, so phosphate group, it is present at the 5' moiety, and at the 3' there is a OH, so hydrogen bond between cytosine and guanosine, that is 3 hydrogen bonds. then 2 hydrogen bond between adenine and guanine. So this is it, like you can see the 5 dash end. So anti-polarity, this is 3 dash. Now adenine, thymine, guanine, cytosine. In here these are the base pairs, and this is sugar phosphate backbone, right? Double DNA double helix. Okay. Now compare the structure of purines and pyrimidines. Can you find out why the distance between the two polynucleotide chains in DNA remains almost constant? So the distance between two polynucleotide chains in DNA remains almost constant. Okay, the proposition of a double helix structure for DNA and its simplicity in explaining the genetic implication became evolutionary. Very soon, Francis Crick proposed a central dogma in molecular biology, which states that the genetic information flows from DNA to RNA and to protein. Okay, so DNA to RNA and to protein. So DNA to DNA, it is a replication. From DNA, from making RNA from DNA is the transcription. Then from mRNA to protein, it is translation. Okay, so this is the central dogma. Now in some viruses the flow of information is in reverse direction, that is reverse transcriptase. Okay, that is from RNA to DNA, that is reverse transcriptase. Okay, can you suggest a simple name to the process? That is reverse transcription. Okay, the packaging of DNA helix. Now DNA is a long polymer, right? Long polymer of polynucleotide, right? Now taken the distance between the two consecutive base pairs, that is 0.34 nanometer. Okay, in meter it is 0.34 into 10 power minus 9 meter, right? If the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pair with distance, how many number of total base pairs in human? 6.6 into 10 power 9 base pairs into the distance between the consecutive base pairs. Okay, that is 0.34 into 10 power 9 meter per base pair. Okay, it comes out to be 2.2 meters. So it is for diploid, and haploid for it is 1.1 meter, right? A length that is far greater than the dimension of a typical nucleus, that is 10 power minus 6 meter. So how is such a long polymer packaged in a cell? Now if the length of the E. coli DNA is 1.36 into 1.36 millimeter, right? This is the length of the, can you calculate the number of base pairs? 4.6 into 10 power 4.6 into 10 power 6 base pair, right? It's given in here, 4.6 into 10 power 6 base pair, right? So 1.33 millimeter is the length of the DNA in the E. coli. Remember this term. In prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. Even if they do not have a defined nucleus, the DNA is not scattered throughout the cell. The DNA, being negatively charged, is held with some proteins that have positive charges in a region termed as nucleoid. Now the DNA in nucleotide is organized in a large loops held by proteins. So DNA in nucleotide is organized in large loops. The DNA in nucleotide is organized in large loops held by proteins, right? So this is the nucleosome. This is the, okay, now we are going to study in eukaryotes. The organization is much more complex. There is a set of positively charged, so there is a set of positively charged basic proteins called histones. A protein acquires charge depending upon the abundance of amino acid residues with charged side chains. Histones are rich in basic amino acid residues lysine and arginines, right? Both the amino acid residues carries positive charges in their side chains. Histones are organized from a unit of 8 molecules called histone octamer. Okay, the negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. A typical nucleosome contains 200 base pair of DNA helix, like base pair of DNA helix. A typical nucleosome contains 200 base pair of DNA helix. Now nucleosomes constitute the repeating unit. Nucleosomes constitute the repeating unit of a structure in nucleus called the chromatin. Okay, so these nucleosomes constitute a repeating unit of a structure in nucleus called chromatin. So thread-like stained color bodies seen in nucleus. The nucleosomes in chromatin are seen as beads on string structure when viewed under the electron microscope. So theoretically, how many such beads nucleosomes do you imagine are present in a mammalian cell? So a typical nucleus contains 200 base pair of DNA, right? The beads on string structure in chromatin is packed is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form chromosomes. So chromatin gets condensed to form the chromosomes. So chromatin fibers condensed to form chromosomes. Now the packaging of chromatin at higher level requires additional set of proteins that collectively are referred to as non-histone chromosomal proteins, that is NHC. So in a typical nucleus, some region of chromatin are loosely packed and stains light and are referred to as euchromatin, and the chromatin that is more densely packed and stains dark are called heterochromatin. So euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. Okay. Now these are the electron microscope picture bits on string, and this is the nucleus, right? And this one is the nucleosome, right? You can see the DNA is wrapped around a histone, 8 molecule of unit, that is histone octamer. This is the H1 histone that links the DNA ends, right? And this is the histone octamer that is made up of 8 molecules, right? 8 molecules. This histone octamer is made up of 8 molecules, right? This is made by 8 molecules. You can see a unit of 8 molecules. To form a unit of 8 molecules, this is the histone octamer right now. The search for genetic material. So transforming principle, then biochemical characterization of transforming principle, and then the genetic material is DNA. It is given by the Hershey and Chase in 1952. So this is we are going to study in here, right? So what about the transforming principle? Transforming principle, which was given by Frederick Griffith. What he worked on Streptococcus pneumonia, which is a bacteria responsible for pneumonia. Okay. Now what he did. So what he did. So so here. So so he worked on the bacteria, that is Streptococcus pneumonia. Now when the Streptococcus pneumonia, that is pneumococcus bacteria, are grown on a culture plate, some now now these are two types of bacteria, right? Some produce smooth shiny colonies, that is yes, but and it is virulent, right? And it is virulent, right? And it is virulent, right? It is poisonous, right? And other produce rough colonies, that is R. This is because the Astran bacteria have a mucus, that is polysaccharide coat, so it protects itself. So now it is virulent, while R strain does not. Mice infected with the S strain virulent die from pneumonia infection, but mice infected with the R strain do not develop pneumonia. Okay, so when the R strain injected into mice die. When R strain injected into mice live. So Griffith was able to kill bacteria by hitting them. He observed that heat killed strain bacteria injected into mice did not kill them. So if the strain is killed and if he injected into mice, then it did not kill. When he strain heat killed injected into mice. So the transforming principle, right? So when the strain is injected into mice, a mice die. Okay, so strain heat kill injected into mice, the mice live. And when the strain heat kill plus R strain live injected into mice, then mice die. Okay, now why this is happen? Like when it took but the strain was killed, it shouldn't be die, but when it put together with the R strain, the mice is die. Okay, now injected into mixture of heat. Okay, but when he injected a mixture of heat kill S and live R bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice. He concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Okay, some transforming principle transfer from the heat killed strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of genetic material. However, the biochemical nature of genetic material was not defined from his experiment. Okay, so the biochemical nature of genetic material was not defined from his experiment. So biochemical characterization of transforming principle. Okay, so the Avery, McLeod and McCarthy, right? So the genetic material was thought to be a protein. So before that, so now Now the transforming principle in Griffith's experiment. So they work on the biochemical nature of transforming principle in Griffith experiment, right? Now they purified biochemicals, proteins, DNA and RNA from the heat killed S cells to see which ones could transform the live cells into the S cells. Now they discovered that DNA alone from the S strain cause R bacteria to become transformed. Now they also discovered that protein digesting enzymes, that protease, and RNA digesting enzyme RNAs did not affect transformation. So the transforming substance was not a protein or RNA. Digestion with DNAs Digestion with DNAs did inhibit transformation, suggesting that the DNA caused the transformation. They concluded that DNA is a hereditary material, but not all biologists were conceived. Can you think of any difference between DNAs and DNAs? DNAs, NAS, it is an enzyme, okay, and DNAs are different types of DNAs, that is bDNA, zDNA. We study the bDNA. The genetic material is DNA. Now Hershey and Chase in 1952, they work with viruses that infect bacteria called the bacteriophages. Now what they did, Hershey and Chase worked to discover whether it was protein or DNA from the viruses that enter the bacteria. Okay, now they grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contain radioactive DNA but not radioactive protein, because DNA contains phosphorus but protein does not. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA, because DNA does not contain sulfur. Okay, so the radioactive phages were allowed to attach to E. coli bacteria. Then as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. So the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Okay, the bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passes from the viruses to the bacteria. Bacteria that were infected with the viruses that had radioactive proteins were not radioactive. This indicates that the protein did not enter the bacteria from the viruses. Now DNA is therefore the genetic material that is passed from the virus to the bacteria. Now you see bacteriophages, they have the radioactive 35US label protein capsule and radioactive 32 phosphorus label DNA. So first step is the infection, second is the blending, that is the removal of the viral coat, and then the centrifugation, that is the separation of the viruses from the bacteria. As you know, in the first case on the left hand side, no radioactive 35S sulfur detected in cells and radioactive 35 sulfur detected in the supernatant, okay, in the slurry format. So this was the protein, right? And in the right hand side, radioactive 32 phosphorus detected in cells, no radioactive detected in supernatant. It means that it is a DNA that been passed. Okay, so the properties of genetic material DNA versus RNA. Okay, so what are the so molecule that can act as a genetic material must fulfill the following criteria. It should be able to generate its replica, that is replication. It should be stable chemically and structurally. It should provide the scope for slow changes, that is mutation, that are required for ovulation. It should be able to express itself in the form of Mendelian characters. Okay, now if one examines each requirement one by one, because of the rules of base pairing and complementarity, both the nucleic acid DNA and RNA have the ability to direct their duplications. The other molecules in the living system, such as proteins, fail to fulfill the first criteria, right? Now also the Griffith experiment shows the stability as the one of the properties of genetic material. Okay, so in line of the DNA, that the two strands being complementary if separated by heating come together. So if you heat, they come together. If separated by heating, they come together. Okay, when appropriate conditions are provided. So further, 2-OH group present at every nucleotide in RNA is a reactive group and makes RNA liable and easily degradable, just because of presence of the 2-OH. Now the RNA is now also known as to be catalytic and hence reactive. Okay, So therefore DNA is chemically less reactive and structurally more stable when compared to RNA. Therefore among the two nucleic acids the DNA is better genetic material. See, in fact, the presence of thymine at the place of uracil also confers additional stability to DNA. So the presence of thymine at the place of uracil also confirms the additional stability to DNA because the chemical name of the thymine is the 5-methyl uracil. Okay, consequently viruses having RNA genome and having shorter lifespan mutate and evolve faster. So viruses having RNA genome and having shorter lifespan mutate and evolve faster, right? So RNA can directly code for the synthesis of proteins, hence can easily express the characters. So RNA can directly code for the synthesis of proteins, hence can easily express the characters, where DNA however is dependent on the RNA for the synthesis of proteins. The protein synthesizing machinery has evolved around RNA. Okay, now all these things indicate that DNA and RNA can function as a genetic material, but DNA being more stable is preferred for storage of genetic information, but for the transmission of this genetic information RNA is better. Okay, now the RNA world. Okay, RNA was the first genetic material. Okay, now why it is that? Because the essential ripe processes such as metabolism, translation, splicing evolve around RNA. Now RNA used to act as a genetic material as well as catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalyst and not by protein enzymes. So RNA used to act as a genetic material as well as catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalyst and not by protein enzyme. This is the ribozyme, right? But RNA being a catalyst was reactive and hence unstable. Therefore DNA has evolved from RNA with chemical modification that make it more stable. But DNA has evolved from RNA with chemical modifications that make it more stable. So DNA being double stranded and having complementary strand further resist changes by evolving a process of repair. Okay, now this is the Watson and Crick model for semi-conservative DNA replication. As you see, now the replication. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, that is as follows: it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick, right? Watson and Crick in 1953. Okay. Now this scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication, right? So Watson Crick model for semi-conservative DNA replication. So the experimental proof. So it is now proven that DNA replicates semi-conservatively. Okay. So but it was first shown in the Escherichia coli and subsequently in higher organisms, such as plants like Vicia faba. Okay, and human cells. The the Matthew Michelson and Franklin Stahl performed the following experiments in 1958. Okay, they grew E. coli in the medium containing N50. Okay, N50 is the heavy isotope of nitrogen. As the only nitrogen source for many generations, the result was that N50 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in cesium chloride density gradient. Please note that N50 is not a radioactive isotope. It can be separated from N40 only based on densities. Then they transferred the cells into a medium with normal N40, like N40 with normal N40, and took samples at various definite time intervals as the cells multiplied. They extracted the DNA that remained in double stranded helices. The various samples were separated independently on cesium chloride gradient to measure the densities of DNA. Can you recall what centrifugal force is and think why a molecule with higher mass density would sediment faster? Okay, Michelson and Stahl experiment. Okay, you see, first they incorporated N50. Okay, this is gravitational force. By gravitational force, now they put it in the medium like N40. So when the N40 is there, so it becomes the hybrid, so N50 and N40. Then after that, after 20 minutes, then after again 20 minutes, means total 40 minutes. So after 40 minutes, right, that is the second generation. It is like, you see, it is like also 1:1, that is light and hybrid. In first case, that is hybrid, light and hybrid, right? So this is it, right? Now the hybrid remains same, like hybrid remains constant, right? In the next as well, there are like two hybrids. Okay, two hybrids and six light, two hybrid and six light, right? Two hybrid and six light, right? um, so two hybrid and six light, right? So this is it. So the medium is in here, like okay, the E. coli was grew in N50, so they can get the N50 as an N50 in their DNA. Okay, so then put it into the N40 medium, right? Then this all happen. So Michelson and Stahl explained that is, so thus the DNA that was extracted from the culture one generation after the transfer from N50 to N40 medium, that is after 20 minutes, E. coli divides in 20 minutes, had a hybrid or intermediate density. The DNA extracted from the culture after another generation, that is after 40 minutes, that is second generation, was composed of equal amounts of this hybrid DNA and of light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the proportions of light and hybrid densities DNA molecule? Light will be one, and no, hybrid will be one, and light will be 7. Okay, so very similar experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome was performed. So VCF-HAVA used the radioactive thymidine on the VCF-HAVA beans by Taylor and colleagues in 1958. Okay, the experiments proved that DNA in chromosome also replicates semi-conservatively. The experiment proved that the DNA in chromosome also replicates semi-conservatively. Now the machinery and the enzymes. So in living cells, that is such as E. coli, the process of replication requires a set of catalysts, that is enzymes. Now the main enzyme is referred to as the DNA-dependent DNA polymerase, since it uses a DNA template to catalyze the polymerization of deoxynucleotides. Okay, these enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. Now E. coli that has only 4.6 into 10 power 6 base pair. Now compare it with human whose diploid content is 6.6 into 10 power 9 base pair. Completes the process of replication within 80 minutes. Okay, E. coli that has only completes the process of replication within 80 minutes, right? That means the average rate of polymerization has to be approximately 2000 base pair per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with a very high degree of accuracy. So any mistake during replication would lead to the mutation. So furthermore, energetically, replication is a very expensive process. Deoxyribonucleoside triphosphates serve dual purposes, as in addition to acting as a substrate, they provide energy for the polymerization reactions. The two terminal phosphates in a deoxyribonucleoside triphosphate are high energy phosphates, as same as in the case of ATP. So deoxyribonucleoside triphosphate DNTP, I think, serve dual purposes. They act as a substrate as well as they provide energy, because the two terminal phosphates in a deoxyribonucleoside triphosphate DNTP are high energy phosphates, as same in case of ATP. So in addition to DNA dependent DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length due to very high energy requirement, the replication occurs within a small Opening of the DNA helix referred to as a replication fork. The DNA-dependent DNA polymerase is catalyzed polymerization only one direction, that is 5' to 3'. This creates some additional complications at the replicating fork. Consequently, on one strand, the template with polarity 3' to 5', the replication is continuous, that is 5' to 3'. But while on the other strand, the template with polarity, that is 5' to 3', then it is discontinued, because it will lead to like 3' to, if it is 5' to 3', then it will lead to like 3' to 5'. Okay, it is discontinuous. The discontinuously synthesized fragments are later joined by the enzyme, that is DNA ligase. The DNA polymerase on their the DNA polymerases on their own the DNA polymerase on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. There is a dependent region of E. coli DNA where the replication originates. Such regions are called as the origin of replication. It is because of the requirement of the origin of replication that a piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a vector. The vectors provide the origin of replication, and this vector provides the replication, and these vectors provide the replication origin of replication. Now, further, not very detail of replication is understood well. In eukaryotes, the replication of DNA takes place at S phase of the cell cycle. The replication of DNA and the cell division should be highly coordinated. So, DNA replication and cell cycle should be highly coordinated, because a failure in cell division after DNA replication results into polyploidy, a chromosomal anomaly. Okay, so this is it, like this is the replication for, you can see, 3' to 5', and when the synthesis goes from 5' to 3', it is continuous, and when 3' to 5', the synthesis goes from 5' to 3', it is discontinuous. Okay, that is 5' to 3'. So the synthesis always takes place from 5' to 3', no 3' to 5'. Okay, now the transcription. Now what is the transcription? We study the replication from DNA to DNA. Okay, now transcription, the process of copying genetic information from one strand of the DNA into the RNA is termed as transcription. Now, okay, the total DNA of an organism gets duplicated. So, however, unlike in the process of replication, which one set in the total DNA of an organism gets duplicated, in transcription only a segment of DNA and only one of the strands is copied into mRNA. This necessitates defining the boundaries that would demarcate the region and the structure and the strand of DNA that would be transcribed. Okay, why both the strands are not copied during transcription? Has the simple answer. The first, if the both strands act as a template, they would be code for RNA molecule with different sequences. Remember, complementarity does not mean identical. Complementarity does not mean identical. And in turn, if they code for protein, the sequence of the amino acids in the proteins would be different. Hence, one segment of the DNA would be coding for two different proteins, and this would just complicate the genetic information transfer machinery. Second, the two RNA molecules, if produced simultaneously, would be to complementary to each other, and hence would form a double stranded RNA. Right, this would prevent RNA from being translated into protein, and the exercise of the transcription would become a futile one. Okay, now the transcription unit. The transcription unit in DNA is primarily by the three regions in the DNA, that is promoter, structural gene, and terminator. So there is a convention in defining the two strands of DNA in the structural gene of a transcription unit. Since the two strands have opposite polarity and the DNA dependent, the RNA polymerase also catalyzes the polarization in only one direction, that is 5' to 3'. As I said, the polarization only takes place in the 5' to 3' direction. Okay, the strand that has the polarity, that is 3' to 5' acts as a template, and that is also known as the template strand. Okay, the other strand which has the polarity, that is 5' to 3', the sequence same as RNA. The other strand which has the polarity, that is 5' to 3', and the sequence same as RNA, except thymine at the place of uracil is displaced during transcription. Strangely, now strangely, this strand which does not code for anything is referred to as the coding strand on all the reference point while defining a transcription until unit is made with coding strand. To explain the point, a hypothetical sequence from a transcription unit is represented below. So you can see 3' to 5', it is a template strand, and then coding strand 5' to 3'. So how can you write So in here, how can you write the sequence of RNA transcript from the above DNA? So you can see the you can see there is like the 3 dash to 5 dash. 3 dash to 5 dash, it is a promoter, and then there is a terminator, and in between there is a structural gene. Okay, so this is the template strand, and below is there is a coding strand. So 5 dash to 3 dash. Okay, so the schematic structure of a transcription unit, right? Now the transcription start site, that is the promoter, right? The promoter and the terminator flank the structural gene in a transcription unit. The promoter is said to be located towards pi dash in upstream of the structural gene. The reference is made with the respect to the polarity of the coding strand. It is a DNA sequence that provides binding site for RNA polymerase. So it is a DNA sequence, and promoter has a DNA sequence, right? And the promoter has a DNA sequence, and the promoter has an is binding site for RNA polymerase, and it is the presence of a promoter in transcription. Okay, so it is a DNA sequence that provides binding site for the RNA polymerase, and it is the presence of a promoter in a transcription unit that also defines the template and coding stands. By switching its position with terminator, the definition of coding and template stand would be reversed. The terminator is located towards 3' end, that is downstream of the coding strand, and it usually defines the end of the process of the transcription. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Some of the properties of this sequence shall be discussed while dealing with the regulation of gene expression. Okay, so translation unit and the gene. So the transcription unit and the gene. Okay, a gene is defined as the structural unit of inheritance. Right? Though there is no ambiguity that the genes are located on the DNA, it is difficult to literally define a gene in terms of DNA sequence. The DNA sequence coding for tRNA or rRNA molecule also define a gene. So it also define a gene, right? Okay, so it also define a gene. However, by defining a cistron as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit could be said to be as monocistronic, mostly in eukaryotes, and polycistronic, that is mostly in bacteria or prokaryotes. In eukaryotes, the monocistronic structural genes have interrupted coding sequences. So in eukaryotes, the monocistronic structural genes have interrupted coding sequences. The genes in eukaryotes the monocistronic structural genes have interrupted coding sequences. The genes in eukaryotes are split. The coding sequences or express sequences are defined as exons. Okay, or express sequences are defined as exons. So exons are said to be those sequences that appear in mature or proceed RNA. Right? The exons are interrupted by introns. The introns or intervening sequences do not appear in mature or proceed RNA. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. So inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Right? Hence, sometimes the regulatory sequences are loosely defined as a regulatory genes, even though these sequences do not code for any RNA or protein. Hence, sometimes the regulatory sequences are loosely defined as a regulatory genes, even though these sequences do not code for any RNA or protein. Sometimes the regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. So types of RNA and the process of transcription. So in bacteria, there are three major types of RNAs: mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, that is ribosomal RNA. So all three RNAs are needed to synthesis a protein in a cell. The mRNA provides the template, tRNA brings the amino acids and reads the genetic code. The RNA plays structural and catalytic role during translation. So there is a single DNA dependent RNA polymerase that catalyzes transcription of all the types of RNA in bacteria. The RNA polymerase binds to promoter and initiates transcription, that is initiation, and this uses nucleoside triphosphates as substrate, and polymerases in a template dependent fashion following the rule of complementarity. It somehow also facilitates opening of the helix and continuous elongation, right? So DNA dependent RNA polymerase also facilitates opening of the helix and continuous elongation. So only a stage of RNA remains bound to the enzyme, and once the polymerase reaches the terminator region, the nascent RNA falls. So the RNA polymerase, this results in termination of the transcription, right? Okay, now this is it. Like, now how is the RNA polymerase able to catalyze all the three steps, which are initiation, elongation and termination? The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with initiation factor sigma and termination factor rho to initiate and terminate the transcription respectively. Okay, in bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation takes place in the same compartment, okay, there is no separation of cytosol and nucleus in bacteria. Many times the translation can begin much before the mRNA is fully transcribed. Consequently, the RNA, the transcription and the translation can be coupled in bacteria. Now in eukaryotes there are two additional complexity. So there are at least three RNA polymerases in nucleus, but in prokaryotes it's only DNA dependent RNA polymerase. So in addition to the RNA polymerase found in the organelles, there is a clear-cut division of label. The RNA polymerase 1 transcribe rRNAs 28S, 88S, then 5.8S, whereas the RNA polymerase 3 is responsible for transcription of tRNA, 5S rRNA and snRNAs, small nuclear rRNAs. The RNA polymerase 2 transcribe precursor of mRNA, the heterogeneous nuclear rRNA, that is hnRNA. The RNA polymerase 2 transcribes precursor of mRNA. The RNA2 transcribes precursor of mRNA, the heteronuclear rRNA, that is hnrRNA. The second complexity is that the primary transcripts contain both the exons and the introns and are non-functional. The second complexity is that the primary transcripts, that is hnRNA, contain both the exons, that is coding or express sequences, and the introns, that is non-coding sequences, and are non-functional. Hence it is subjected to a process called splicing, where the introns are removed and the exons are joined in a definite order. Now hnRNA undergoes additional processing called as capping and tailing. So hnRNA undergoes additional processing called as capping and tailing, right? In capping, an unusual nucleotide, that is methyl guanosine triphosphate, is added to the 5' end of hnRNA. So this is capping, and in tailing, adenylated residues, that is 200 to 300, are added at 3' end in a template independent manner, right? So it is fully processed hnRNA, now called the mRNA, that is transported out of the nucleus for the translation. Okay, now the significance of such complexities is now beginning to be understood. Now the split gene arrangements represents probably an ancient feature of the genome. So this split gene arrangement in the eukaryotes, it shows like it's like monocystonic, right, and polycystonic in prokaryotes. So this shows like, so it is fully proceed the presence of introns. Now the presence of introns is reminiscence of antiquity, and the process of splicing represents the dominance of RNA world. So okay, now the genetic code. So we are going to study the genetic code. So what about the genetic code? It is triplet, right? First one thing is it is triplet, right? So it is triplet, and it was George Gamow, a physicist, who argued that since there are only 4 bases, and if they have to code for 20 amino acids, the code should be constitute a combination of bases. Okay, now he suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. Okay, Now this was very bold proposition because the permutation combination 4 power 3 would be generate 64 codons. Okay, so 4 power, if 4 power 2 it generate only 60 codons. Okay, but we have the 20 amino acids, but we need 20 amino acids, right? So to make it like, so it would be like generate 64 codons, so it is 64 codons generating many more codons than required. Okay, now the chemical method developed by Hargobin. So okay, the codon was triplet. Okay, the chemical method developed by Hargobin Khurana was instrumental in synthesizing RNA molecules with defined combinations of bases, homopolymers or copolymers. Okay, I repeat it again. The chemical method developed by the Hargobin Khurana was instrumental in synthesizing RNA molecules with defined combination of bases, that is homopolymers and copolymers. Marshall Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. Several Okao enzyme polynucleotide phosphorylase was also helpful in polymerizing RNA with defined sequences in a template independent manner, that is enzymatic synthesis of RNA. So several Okao enzyme polynucleotide phosphorylase was also helpful in polymerizing RNA with a defined sequence in a template independent manner, enzymatic synthesis of RNA. Okay, now the codons for various amino acids. You see the first position, then second position, then third position. UCAG, 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 and all these. Okay, the silent features of the genetic code. Don't remember this table. The silent feature of the genetic code as follows. The codon is triplet. 61 codons code for amino acids and 3 codons do not code for any amino acids. Hence they function as a stop codon. Okay. Now some amino acids are coded by more than one codon. Hence the code is degenerate. It is degenerate, right? Because the some amino acids are coded by more than one codon. Hence the code is degenerated. The codon is read in mRNA in a contiguous fashion. There are no punctuation. Okay, the code is nearly universal. For example, from bacteria to humans, U would code for phenylalanine. Some exceptions to this rule have been made in mitochondrial codons and in some protozoans. Now, AUG has dual functions. It codes for methionine mate and it also acts as initiator codon. Now, UAA, UAG, and UGA are stop terminator codons. So, if following is the sequence of nucleotides in mRNA, predict the sequence of amino acid coded by it. Predict the sequence of amino acids by it. So AUG is a methionine, UUC, UUU is a phenylalanine, right? Phenylalanine. Then UUC is also phenylalanine. UUC is also phenylalanine. Okay, UUC is also phenylalanine. UUC is also phenylalanine. Remember this, right? Now in here it is given like methionine and then all the phenylalanine. Okay, do you face any difficulty in predicting the opposite? Can you correlate it which two properties of genetic code you have learned, right? Now the mutations and genetic code, right? So mutation and genetic code. Okay, point mutation is a change of single base pair in gene for beta globin chain that results in the change of amino acid residue, that is glutamate to valine, that is GAG to GUG. Now this is known as a sickle cell anemia, right? So effect of point mutation that inserts or deletes a base in structural gene can be better understood by, as you know. So the conclusion of all this exercise is insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. However, such mutations are referred to as frame shift insertion or deletion mutations. So frame shift insertion or deletion mutation. Okay, so all this is known as the frame shift insertion or deletion mutation. Insertion or deletion of three or its multiple bases insert or delete in one or multiple codon. So insertion or deletion of three or its multiple bases. So insertion or deletion of three or its multiple bases insert or delete in one or multiple codon. Hence one or multiple amino acids and reading frame remains unaltered from that point onwards. So insertion or deletion of 3 or its multiple bases does not changes, it's unaltered from that point onwards. Okay, now the tRNA, that is adapter molecule. So the tRNA, the adapter molecule, right? So mainly it was given by the Crick. Okay, now an adapter molecule that would on the other hand read the code and on the other hand would bind to a specific amino acids. Now tRNA then called as sRNA, that is soluble RNA, right? Okay, now this is the tRNA. tRNA has an anticodon loop that has bases complementary to the code, and it also has an amino acid acceptor. It also has an amino acid acceptor end to which it binds to the amino acids, and tRNAs are specific for each amino acids. For initiation, there is another specific tRNA referred as initiator tRNA, but there are no tRNAs for stop codon. Important: the secondary structure of tRNA has been depicted that looks like a clover leaf, but the actual structure tRNA compact, it looks like inverted L. Now this is the tRNA, that is the adapter molecule. On downside, it is mRNA that has a codon and anticodon loop present on tRNA. It has 5' end, it has 3' end. At 3' end, it has the amino acid acceptor end. Okay, so this is it. So AGU has an anticodon UCA, right? UCA. So this is the tRNA, right? And UAC has AUG. So this can be asked. Okay, so codon and anticodon. AGU has anticodon UCA and UAC has anticodon AUG, right? So this is it. Now the translation. So translation refers to the process of polymerization of amino acids to form a polypeptide. So translation refers to the process of polymerization of amino acids to form a polypeptide. Okay, the order and sequence of amino acids are defined by the sequence of bases in the mRNA. The order and sequence of amino acids are defined by the sequence of bases in mRNA. The amino acids are joined by a bond which is known as a peptide bond. The amino acids are joined by a bond which is known as a peptide bond. The formation of a peptide bond requires energy. Therefore, the first step itself, amino acids are activated in presence of ATP. Therefore, in the first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called as charging of tRNA or amino acylation of tRNA, to be more specific. So charging of tRNA or amino acylation of tRNA. So this is the first step, right? This is the first phase itself. Okay, if two such charged tRNA they brought enough they for the peptide bond. Okay, and this is energy requiring process. The presence of a catalyst would enhance the rate of peptide bond formation. Okay, the cellular factory responsible for synthesizing proteins is the ribosome. The ribosome consists of a structural RNAs and about 80 different proteins. In its inactive state, it exists at two subunits, a large subunit and a small subunit. So when the small subunit encounters an mRNA, the process of translation of mRNA to protein begins. Okay, there are two sites in the large subunit for subsequent amino acids to bind to and thus be close enough to each other for the formation of peptide bond. The ribosome also acts as a catalyst, that is 23S. RNA in bacteria is the enzyme, that is ribozyme, for the formation of peptide bond. Okay, a translation unit in mRNA is the sequence of RNA that is flanked by a start codon AUG and the stop codon and codes for a polypeptide. So a translation unit in mRNA is the sequence of RNA that is flanked by a start codon, that is AUG, and the stop codon and codes for a polypeptide. An mRNA also has some additional sequences that are not translated and are referred to as untranslated region. Now UTRAs are present at both 5' end before the start codon and 3' end after the stop codon. They are required for efficient translation processes. As you can see, it is shown before the start codon and after the start codon it is UTR, untranslated regions, right? They are used for to efficient this process. Then you can see it is mRNA that has codon, and then there is small subunit of RNA is attached in large subunit having having two sides, right? In last subunit it is growing polypeptide chain is tRNA. As you can see, alanine is attached to valine, so tRNA GCA CGU and GUU, that is CAA. Now UUA is coming and AGG is coming, UCC. So this is the translation, right? Now for initiation, the ribosome binds to the mRNA at the start codon AUG, that is recognized by only the initiated tRNA. The ribosome proceeds to the elongation phase of the protein synthesis. During this stage, the complex is composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. The ribosome moves from the codon to codon along the mRNA. The ribosome moves from codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequences detected by the DNA and represented by the RNA. Very important. It is detected by the DNA, translated into polypeptide sequences detected by the DNA and represented by RNA, and at the end a release factor binds to the stop codon and terminating translation and releasing the complete polypeptide from the ribosome. Now regulation of gene expression. So we are going to study the lac operon. Okay, so what's in the lac operon? So regulation of the gene expression refers to a very broad term that may occur at various levels. Okay, considering that gene expression results in the formation of polypeptide, it can be regulated at several levels. In eukaryotes, like in eukaryotes, the regulation could be exerted at like transcription level, that is formation of primary transcript, and processing level, that is regulation of splicing, that is the removal of the introns, and then there is the transport of mRNA from nucleus to the cytoplasm. Then there is there is a transport of mRNA from nucleus to the cytoplasm, and at the fourth, that is the translation level. So this is it, like, okay. So the genes in a cell are expressed to perform a particular function or a set of functions. So if an enzyme called the beta-galactosidase is synthesized by an E. coli, it is used to catalyze the hydrolysis of a disaccharide, that is lactose into galactose and glucose. So disaccharide, lactose into galactose and glucose enzyme is beta-galactosidase in E. coli, okay. And the bacteria use them as a source of energy, okay. So hence, if a bacteria do not have lactose around them to be utilized for the energy source, they would have no longer require the synthesis of the enzyme beta-galactosidase. Therefore, in simple terms, it is a metabolic, physiological, environmental condition that regulate the expression of the genes. The development and differentiation of embryo into adult organism are also result of the coordinated regulation of expression of several sets of the genes, okay. In prokaryotes, the control of the rate of the transcriptional initiation is the predominant site for the control of gene expression. So in a transcription unit, the activity of RNA polymerase at a given promoter is in the turn regulated by interaction with the accessory proteins, which affects its ability to recognize start sites. So these regulatory proteins can act both. Okay, so the regulatory proteins can act both positively, that is activators, and negatively, that is repressors. The accessibility of the promoter regions of the accessibility of the promoter regions of the prokaryotic DNA is in many cases regulated by the interaction of proteins with sequence term operators, okay. By interaction of proteins with sequences term operators, okay. The accessibility of promoter regions of prokaryotic DNA is in many cases regulated by the interaction of proteins with sequences term operators. The operator region is adjacent to the promoter elements in most operands, and in most cases the sequence of the operator bind or repressor protein, okay. Each operand has its specific operator and specific repressor. For example, lac operator is present only in the lac operon. It interacts specifically with the lac repressor only, okay. Now we are going to study the lac operon. So the elucidation of the lac operon was also a result of a close association between a geneticists Francis Jacob and biochemist, that is Jacob Monod. They were the first to illustrate a transcriptionally regulated system. In lac operon, here lac refers to the lactose. A polycystonic structural gene is regulated by a common promoter and a regulatory genes. Such arrangement is very common in bacteria and referred to as operon. To many few examples like operon, trip operon, arac operon, his operon, val operon are present. Okay. The lac operon consists of one regulatory gene, that is the I gene. Here the I does not refer to inducer, rather it is derived from the word inhibitor. So I is not inducer, it is the inhibitor, right? It is derived from the inhibitor. And the three structural genes Z by A, the I gene codes for repressor of the lac operon, the Z gene codes for beta galactosidase beagle, or which is primarily responsible for the hydrolysis of the disaccharide lactose into its monomeric units, that is like galactose and glucose. The Y gene codes for a permease which increases the permeability of the cell to beta galactosidase, and the A gene encodes a transacetylase. Hence, all the three gene products in lac operon are required for the metabolism of lactose. In most other operons as well, the genes present in operon are needed together to function in a same or related metabolic pathway. Now, in the absence of inducer, you can see that the I makes the repressor mRNA, and repressor binds to the operator region and prevents the RNA polymerase from transcribing the operator. Okay, at the promoter region the RNA polymerase binds. Okay, but in the presence of inducer, when there is an inducer, that is now inducer can be the, now it's given in here, the you can see okay, lactose is a okay, so I first read the text. Lactose is the substrate for the enzyme beta galactosidase, and it regulates switching on and off of the operon. So lactose is the substrate, right? Hence it is termed as the inducer. Now lactose, it is a inducer, right? In the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cells through the action of permease. Remember, a very low level of excretion of lac operon has to be present in the cell all the time, otherwise lactose cannot enter the cells. The lactose gene induces the operon in the following manner. The repressor of the operon is synthesized all the time consequently from the I gene. Okay, the repressor protein binds to the operator region of the operator and prevents RNA polymerase from transcribing the operon. In the presence of an inducer such as lactose or allolactose, the substrate can be lactose or allolactose. The repressor is inactivated by interaction with the inducer. So the repressor is inactivated by interaction with the inducer. Okay, so inducer, that is lactose. So the repressor is inactivated. So the repressor is inactivated by the interaction with the inducer. Okay, this allows RNA polymerase access to the promoter and transcription proceeds. Okay, so in presence of lactose, right, in presence of lactose, that is inducer, it is the repressor gets inactivated. The repressor gets inactivated. Okay, so this is like, okay, so essentially regulation of lac operon can also be visualized as regulation of enzyme synthesis by its substrate. So if the substrate is present, then there is a synthesis of the enzyme, that this substrate, that is inducer, it what it does, it inactivates the inducer, it inactivates the repressor. Here is given here in the presence of an inducer, that is lactose allolactose, the repressor is inactivated by interaction with the inducer, right? Now remember glucose or galactose. Now the glucose and galactose cannot act as inducers for the operon. Can you think for how long the lac operon would be expressed in the presence of lactose until it's needed? Regulation of lac operon by repressor is termed as negative regulation. Okay, lac operon is under control of positive regulation as well. Okay, now the human genome project, it is the biggest project. Human genome project was called a mega project, and it approximately, the human genome is said to have approximately 3 into 10 power 9 base pairs, and if the cost of the sequencing required is $3 per base pair, the estimated cost in the beginning, the total cost, estimated cost of the project would be approximately, that is $9 billion US. Okay, $9 billion US, right? Now HEB was closely associated with the rapid development of new era in biology called the bioinformatics. high-speed computational devices for data storage and retrieval and analysis. Goals of HGP, Human Genome Project. Some of the important goals of HGP were as identify all the approximately two. Okay, these goals are not important, but to address the ethical, legal, social issues that may arise from the project. The Human Genome Project was a 30-year project coordinated by the US Department of Energy and National Institute of Health. During the early years of the HGP, the Wellcome Trust UK was a major partner. Additional contributions came from Japan, France, Germany, China, and others. The project was completed in 2003. Okay. Now there are some non-human organisms uh, that is also made. Many non-human model organisms such as bacteria, yeast, Caenorhabditis elegans, a free living non-pathogenic nematode, Drosophila, the fruit fly, plants, rice, and Arabidopsis have been sequenced. Now methodologies. The methods involve there are two approaches. One approach focuses on identifying all the genes that are expressed as RNA, referred to as explain sequence tags. The other took the blind approach of simply sequencing the whole set of genome that contain all the coding and non-coding sequence and later assigning different regions in the sequence with functions. A term referred to as the sequence annotation. Okay, for sequencing the total DNA from a cell is isolated and converted into random